Today I'm going to explain about Kreb cycle. Kreb cycle known as citric acid, a biochemical pathway used to generate energy through oxidation of acetyl CoA. Its synthesis of NADH and amino acid. It occurs in mitochondria matrix. In the Kreb cycle, the first biomolecule we are going to see is pyruvate. Pyruvate is 3-carbon molecule. Pyruvate derives from glucose 6-carbon molecule through the process of glycolysis. Pyruvate oxidized to acetyl CoA, which is 2-carbon molecule. Carbon dioxide and NADH is generated from the Kreb cycle. Acetyl CoA is combined with oxaloacetate through the process of condensation to produce citrate, which is 6-carbon molecule. Citrate rearranges by the removal of water molecule and the addition of water to a different carbon atom to form isocitrate, which also 6-carbon. The reaction is catalyzed by aconitase. Isocitrate is oxidized to form oxalosuccinate, which is 6-carbon molecule. NAD+ is reduced to become NADH. This is catalyzed by isocitrate dehydrogenase. Oxalosuccinate, which is still bound to the enzyme of isocitrate dehydrogenase, is unstable. It immediately undergoes decarboxylation, which loss of carbon dioxide, and it converted to alpha-ketoglutarate, which is 5-carbon molecule. Oxidative decarboxylation of alpha-ketoglutarate take place. This produces succinyl coenzyme A, which is 4-carbon. NAD+ is reduced to become NADH and is forming succinyl coenzyme A, which is 4-carbon molecule. The next step is substrate level phosphorylation take place. Succinyl coenzyme A is converted to succinate, which is 4-carbon. The energy release is used for phosphorylation of GDP, which is converted to GTP. This reaction is catalyzed by succinyl coenzyme A synthesis. Succinate is oxidized to fumarate, which is 4-carbon. 2 hydrogen atoms are transferred to FAD to form FADH2. This reaction catalyzed by succinate dehydrogenase. Fumarate becomes dehydrated by addition of water, then converted to malate, which is 4-carbon. This reaction is catalyzed by fumarase. Malate is oxidized to regenerate oxaloacetate, which is 4-carbon. NAD+ is reduced to become NADH+. The reaction is catalyzed by malate dehydrogenase. Oxaloacetate can be used to combine with acetyl CoA and the cycle is repeated again. The result of Kreb cycle, it will produce 3 NADH, 1 FADH2, 1 GTP and 2 carbon dioxide. Since the Kreb cycle repeat 2 times, it will produce 6 NADH, 2 FADH2, 2 GTP and 4 carbon dioxide. The product of Kreb cycle will transfer to electron transport chain to generate the ATP.
In mitochondria, energy is stored in NADH and FADH2. Glucose oxidized during glycolysis and Kreb cycle, and it produces coenzyme NADH and FADH2. So, inside mitochondria matrix, the NADH will transfer its electron to coenzyme Q, while the proton will pass through the NADH dehydrogenase to intermembrane space, and NADH will convert to NAD+. And then, the electron will transfer to BC1 complex. FADH2 will transfer the 2 electrons to coenzyme Q. And then the 2 proton will transfer out to the intermembrane space. The electron will transfer to the BC1 complex and the FADH2 will turn to FAD. The electron from cytochrome C will transfer to cytochrome C oxidized complex. The proton at the intermembrane space will enter the mitochondrial matrix through the ATP synthase. At the same time, the proton will bind to the ADP and phosphate, and it will change to ATP. Glycolysis comes from word glyco, which means glucose, and lysis, breakdown. So overall, glycolysis is the breakdown of glucose in cytosol to produce energy. One molecule of glucose, which is a 6-carbon compound, will be broken down into 2 molecules of pyruvate, which is a 3-carbon compound. In this conversion, energy is released in the form of ATP and NADH. The pyruvate form is further utilized in various energy-producing pathways that produce more energy. Now, let's look at glycolysis reaction. This begins with a molecule of glucose that enters the cytosol, and the first reaction that takes place is conversion of glucose to glucose 6-phosphate. The enzyme that catalyzes this reaction is hexokinase. The hexo means 6, denoting 6-carbon compound glucose, and kinases is a group of enzymes that catalyze phosphorylation. Hexokinases transfer a molecule of phosphate from ATP to glucose, converts it to glucose 6-phosphate. In this reaction, ATP is converted to ADP. Thus, it utilizes 1 ATP. Next reaction, glucose 6-phosphate is converted to fructose 6-phosphate, which catalyzes by isomerase enzyme. In the next step, fructose 6-phosphate is converted to fructose 1,6-biphosphate. In this reaction, another phosphate group is added. This reaction is catalyzed by phosphofructokinase. In this reaction, another ATP is converted to ADP, thus it utilizes another molecule of ATP. Next, fructose 1-biphosphate is split into 2 molecules, which are glyceraldehyde 3-phosphate and dihydroexciton phosphate. These molecules are 3-carbon compounds, and the enzyme that catalyzes this reaction is called aldolase. This reaction composes of the first phase of glycolysis, where energy is consumed. The 2 compounds are actually isomers. Thus, this reaction can be interconverted by isomerase enzyme. Bear in mind that in the second phase, every reaction happens twice and produces twice of products. The reaction furthered by glyceraldehyde 3-phosphate, which is converted to 1,3-biphosphoglycerate. In this reaction, NAD+ is converted to NADH, and an addition of inorganic phosphate is added. This reaction is catalyzed by enzyme dehydrogenase. Next, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. In this reaction, 1 ADP is converted to ATP. In this reaction, it generates 1 molecule of ATP. 3-phosphoglycerate is then converted to 2-phosphoglycerate by enzyme phosphoglycerate mutase. 2-phosphoglycerate is then converted to phosphoenol pyruvate by the enzyme enolase, and in this reaction, magnesium ion is utilized. In the final step, phosphoenol pyruvate is converted to pyruvate by the enzyme pyruvate kinase, and another ADP molecule is converted to ATP molecule, which another ATP molecule is generated. This composes the second phase of glycolysis. In this second phase, the energy is actually produced. Therefore, that reaction shows that 1 glucose plus 2 molecule of ADP plus 2 molecule of inorganic phosphate plus 2 NAD+ will produce 2 pyruvate plus 2 ATP plus 2 NADH.